Хорошо. Дима, разрешите я сейчас проверю, будет листаться или не будет, пока у меня что-то не листается. Ну, это кнопкой вправо, влево, что-то не листается. Мышкой по презентации. Так. О, мышкой по презентации оказалась плодотворной идеей. Да, все нормально. Сейчас уже вправо-влево есть, да. Хорошо, я могу начинать? Спасибо, Дмитрий. Благодарю, я начинаю. Угу. Спасибо. Good afternoon, ladies and gentlemen. Topic of today's lecture is immune mechanisms of the inflammation, first topic. Second, innate immunity of the body. Third, primary recognition of non-self and damaged self. By the way, this primary recognition of non-self and damaged cells is commented by the drawing in the right lower part of our slide. What here we see how gray lymphocyte, our protective cell, enjoys the finding of the self cell. Other lymphocyte that is drawn in the right direction is angry because he is a non-self bacterium. It means this recognition of non-self and damaged self, this is a basis of all the immunology. The next sub-question, acquired immune response. Acquired. So in this response, subtopics, antigens and their characteristics, processing and presentation of antigens. Now let's start from the review of the immunology, very, very brief. So all the immunity, our immunity, is the state of protection against genetically different substances or organisms. All the molecules that have the fingerprint of the foreign genome, not our genome, is considered as the non-self or foreign, and this is a substance that must be removed from the human body. Immune system is directed for maintaining of the stable and clean state of our organism without foreign molecules. But in the real life, this is impossible. All the environment is filled with the foreign microbes. It means immune system is switched on in any moment of our life. Immune system and immune responses are subdivided in two large parts. One is named innate immunity. What in this slide you see in the left part, some of the innate immunity cells and molecules, and acquired or adaptive immunity that is shown by the square in the right part of this slide. Innate immunity, this is the immunity that is maintained by inborn structures and inborn processes. Innate immunity occurs in any moment of our life, and innate immunity is comparatively low specific. Innate immunity is directed for prevention of the entering and spreading of the foreign microorganisms of their products in our body. Innate immunity, take a look to this slide, consists of the barriers. Barriers formed by epithelial cells and skin cells, keratinocytes. Here you see how the star-like pathogens may try to penetrate these barriers. Below you see three cells, phagocytotic cells. All the professional phagocytotic cells are cells of the innate immunity. What the cells do you see here? Phagocytes, purple neutrophil, and stained green macrophage. And at the right direction from these cells, so-called dendritic cell that has many processes to contact at the same time to the many other immune cells that must be informed by the dendritic cell about presence of the foreign molecules. In the lowest part of the innate immunity, figure characterizing this are plasma proteins. Plasma proteins here shown as a yellow object of different shape. Plasma proteins, this is a very variable group of the molecules that have a different mechanism of activity, altogether act to eliminate foreign substances, foreign organisms, microbes, viruses, and tumor components from our cell. And the cell, yellow cell that is shown in the low part of the innate immunity mechanism is NK cell. This is a natural killer cell that kills virus-infected or bacteria-infected human cells to kill the cells that serve as the factories of production of the pathogens. You see that all the innate immunity components and reactions in their kinetics of development need hours for the development. But here the timetable maximum marker here is 12 hours. In the contrast, adaptive or acquired immunity processes, they are associated to very long, rather long period of the activation. You see that adaptive immunity mechanisms require days and weeks for their development. You see here lymphocytes. Adaptive immunity is based on the activity of lymphocytes and antibodies. The upper lymphocyte among the adaptive immunity mechanisms is of the blue color. You see here that this blue cell has the blue Y-shaped receptors of the membrane, the immunoglobulin receptors, recognizing antigens. Star-like virus reacts to one of the receptors of the blue cell here. After this recognizing of the antigen by the receptor of lymphocyte, this lymphocyte start to replicate. Proliferation is happening. This lymphocyte serves as the maternal cell for appearance of the thousands, millions cells of the progeny lymphocytes. All of them have the same antigenic characteristic. It means their receptors are the same. All of these B lymphocytes, of course, B lymphocytes, newly formed B lymphocytes, transform to the plasma cells that don't replicate. These cells are not shown here. They live 2 days. During that, these plasma cells produce very high amounts of the antibodies, free antibodies, soluble antibodies. These antibodies find all the microbes that have antigen that was recognized before this by the first B cell. They, in this way, label this antigen, they attack to this antigen immune cells and activate complement system and other instruments, and finally these antibodies serve as the molecules that may remove antigens. Lower you see how yellow cells are developed. Yellow cell shown here, this is a T cell. T cell is a lymphocyte that may occur in form of the T cytotoxic killer cells, so-called CD8 cells, or T helper cells, CD4 cells. With this lymphocyte shown here, yellow, maybe one of these cells. You see that this lymphocyte contracts to the green macrophage that presents foreign antigenic determinant peptide to the specific receptor of the T cell. After the T cell again start to proliferate, many cycles of replications happen. These T lymphocytes serve as the maternal cell for millions of the T lymphocytes of the same specificity. After these T lymphocytes again contact to the macrophages that may activate newly formed T helpers for synthesis of the cytokines that stimulate immune responses. If this yellow cell shown in the right part of the slide is T killer cell, CD8 cell, The cell recognizes foreign antigens on the surface of macrophage if this macrophage is infected is itself, and the killer cell kills this macrophage. What are the important difference between innate immunity and adaptive immunity? In the innate immunity, protection may be switched on without replication of cells. Of course, cells will replicate, but the first line of our defense doesn't require replication as a condition for beginning of the reaction. In the case of the adaptive immunity, situation is absolutely different. Initially, few cells that have the specific receptors for the certain antigen must proliferate many times, replicate, and only progeny cells of this first line cells that recognize antigen may really influence to the fate of humans, may really maintain the protection. Proliferation requires time because cells replicate during a certain time, and time required is measured by days and weeks. It means innate immunity reactions are very quick but low specific. Adaptive immunity reactions are slow but highly specific and very highly powerful. Inflammation. Inflammation, you know that this is a reaction for the damaging of infection. But here you see a photograph of the boy with a red and swelled cheek. Inflammatory processes happen here. But inflammation is the sum of the changing that occur in tissues due to the injury or infection. Inflammation, this is a universal protective reaction of our organism that is developed as a response for almost all the pathogens that are known. Inflammation clinically associated to the redness of the tissue, swelling, warmth, and pain. In the right part of the slide, you see what gray root bacteria invite tissues and from the vessels, what red vessels, to the infected area, transported blue cells and neutrophils that attracted and accumulate in the infected area together with neutrophils macrophages, what purple cells, the lowest in this figure, engulf bacteria and produce the cytokines to stimulate inflammation. A green cell upper is basophil or mast cell producing histamine. If the cell is activated by beginning of the inflammatory process, histamine increases the blood flow in this area, increases vascular permeability for blood plasma and for cells that circulate in the plasma. This water and plasma from vessels accumulate in tissues, help migrating bacteria to be accumulated, infiltrate these tissues, and destroy bacteria. But here in the painting in the upper right part of the slide, you see that inflammation, first of all, depends on the increasing of the blood flow in the place of infection. So the signs of inflammation are due to the increased blood flow, increased capillary permeability, and escape of fluid and cells into tissue spaces. Inflammation due to infections often starts with the activation of the innate immunity mechanisms like complement system, their soluble proteins in the blood. This process causes the production or release of the mediators of inflammation. What one mediators of inflammation is histamine secreted by mast cell. Other mediators of inflammation, for example, interleukin 1, interleukin 12, interleukin 6, are produced by macrophages, and they also enhance the vascular permeability, increasing. Inflammatory process is a protective process in our body may be subdivided into the certain steps, at least some 7 steps. First step is beginning of inflammation depends on the primary recognition of non-self and damaged self cells by innate immune mechanisms. Innate immunity, this is the first part of immunity that reacts for the foreign entities. What do you see here? You see innate immunity activation, first step, beginning of inflammation. Here you see damaged tissues colonized by the gray bacteria. First, from the top, you see blood platelets in the tissues that react to the bacterial substances, producing proteins of the clotting system, blood clotting proteins that form the barrier to prevent spreading of the bacteria in the death of tissues. Blood platelets substances, some of them may stimulate release from mast cells of the histamine. Histamine causes intensification of blood flow in the subcutaneous capillary system, stimulates increase of permeability for liquid, and liquid from the vessels is transported to the infected tissues, and vascular permeability, when it's enhanced, stimulate migration of the migrating lymphoid cells from vessels to the infected tissues. Among these cells, neutrophils, basophils, macrophages, lymphocytes, fibroblasts, and some other cells. Here you see blue neutrophils that engulf bacteria and digest them. You see below macrophage, purple cell, that also digests bacteria, but at the same time produce cytokines, for example, interleukin-1, tumor necrosis factor, that additionally increases the blood flow and recruiting of the migrating lymphoid cells from vessels to the infected area. And macrophages start a process that is named presentation of antigen for lymphocytes, for T lymphocytes, presentation for T lymphocytes. This process is not shown. Second step is name of the inflammatory response is processing and presentation of antigen, and secondary recognition of non-self. What in the first slide devoted to inflammation, you see that immune cells engulf and digest bacteria. Why? Because they have special receptors, so-called pattern recognizing receptors that recognize foreign substances on the surface of bacteria. These surface substances are named pathogen-associated patterns. It means foreign molecules that are recognized by innate immunity cells with these foreign substances is named pattern. Pathogen-associated pattern, it means bacteria-associated pattern, but not antigen. These patterns and their recognition is low specific. In the contrast, secondary recognition of non-self. This recognition of not the pathogen-associated patterns, but of antigens. What are the difference between antigens that are different and pathogen-associated patterns that are also different? Pathogen-associated patterns are usually small molecules, smaller than antigens, that can cause very specific recognition reaction. Patterns are used usually for differentiate self from non-self without details what the non-self now is present in the human body. Antigens are very highly specific foreign entities. Antigens are larger than patterns. Antigens may be recognized by antibodies and T cell receptors with a very high specificity, and acquired immunity cells and acquired immunity immunoglobulins, they differentiate antigen of one bacteria from antigen of other bacteria, very highly specific. So this is the difference between primary and secondary recognition of non-self. Primary is low specific, and secondary is very highly specific. What the process is shown here. In the left part of the drawing, you see very large yellow cell with a very long processes. This is a antigen-presenting cell, namely dendritic cell. Dendritic cell has the pattern recognizing receptors that recognize foreign molecules on the surface of the bacterium. Bacterium is shown by the green oval that is engulfed and phagocytosed by dendritic cell. After that, this phagocytosed cell is undergone to the digestion when phagosome is fused to the lysosome. Lysosomal enzymes destroy compounds of the bacterium. Bacterium is disrupted. From these disrupted bacteria, antigenic determinants are released. They are minimum molecules with a specific characteristic that may be recognized by antibodies and other molecules of the acquired immunity system. These antigens are shown as the small green ovals. These green ovals, small antigenic bacterial determinants, are hybridized, usually interact with the MHC2 proteins. MHC2 proteins are shown as the green molecules with the incision, and this incision binds antigenic molecules, antigenic peptides. And you see how these complexes of MHC2 molecule to antigenic peptides are transported to the surface of the dendritic cell, surface of one of the processes, and displayed to the T lymphocytes on the surface of this antigen-presenting cell. One word about MHC proteins. MHC, it means major histocompatibility complex, major histocompatibility complex type 2. Major histocompatibility complex type 2 molecules are the markers of self-origin of these molecules that occur on the surface of all the antigen-presenting cells. If the certain cell doesn't have MHC2 protein, you can say that this is another antigen-presenting cell. If you find MHC2 molecules on the cell, you understand this is antigen-presenting cell. Antigen-presenting cells are dendritic cells, macrophages, monocytes, and B lymphocytes. What does it mean, antigen presentation? Antigen presentation is the result of the complexing of the MHC2 proteins to the bacterial antigenic peptides in the Golgi apparat, and the transportation of these complexes to the surface of the antigen-presenting cell, and presentation of the MHC2 protein combined to the bacterial antigen to the T cells. In the upper right part of the slide, you see that MHC2 molecule contains yellow antigenic peptide that reacts to the T cell receptor of this T lymphocyte, and T cell receptor, if fits to this bacterial antigen, stimulates this T lymphocyte for proliferation, for replication of these cells. Not any T lymphocyte may recognize this presentation. Only T lymphocyte that has CD4 receptor. CD4 receptor shown here as a blue rod on the surface of this T lymphocyte. This CD4 receptor binds to the MHC2 and confirms that this MHC2 is a part of the antigen-presenting cell that is a cell of the same human personal organism. It means, what does it mean, histocompatibility complex? This is a complex of proteins that serve as the marker of self-origin of cell that carries this MHC. Well, antigen presentation, if it happens, causes stimulation of the replication of this T lymphocyte, producing of progeny of T lymphocyte, and maturation of this progeny into one of the types of the immunocompetent cells. Immunocompetent, it means cells that may serve as a matured cell maintain visualization of the function of this cell. After secondary recognition of non-self, adaptive acquired immunity response is developed. This is the third stage. What development of acquired immunity response, this is a process of replication of activated B cells activated by antigen and activated by antigens T cells, and the synthesis of the antibodies, beginning of this process. This process is followed by accumulation of immunocompetent cells of these mature T cells, mature B cells, plasma cells producing antibodies, and antibodies at the site of infection and injury, or accumulation at the place infected by the organism. Pay attention to this drawing that you see. What do you see? In the upper right part, you see stage number 2, processing and presentation of antigen. This is step number 2. What do you see below in the green square? You see the beginning of the cellular immune response. You see star-like yellow cell with almost black nucleus. This is a dendritic cell, you may recognize, because processes are very long. This dendritic cell is antigen-presenting cell. This dendritic cell has pattern recognizing receptors for non-self molecules. This receptor is shown as a red surface molecule of the dendritic cell. This red molecule recognizes and binds to the purple bacterium, flagellated bacterium that's shown here. What has happened after that? After that, this bacterium is engulfed, is digested, antigen is released from this bacterium, and antigen is processed. It means antigen peptides of this bacteria are combined to the MHC proteins. These antigens may be combined to MHC2 proteins, that is shown in the one of the left part of this dendritic cell. Or this antigen may be combined to the MHC1 protein, also major histocompatibility complex protein type 1, that is shown in the one of the processes of the right part of the dendritic cell. MHC2 molecule form the complex with the antigenic peptide, shown here the blue oval, and this peptide may stimulate T cells, T helper cells, that have the T cell receptors specific for this antigenic peptide. If the certain helper cell has TCR that has no specificity for this antigen, this T helper cell will ignore presence of this presentation. If this T helper cell will have TCR specific T cell receptor for the antigen, T helpers will be activated. Of course, activated after binding of CD4 receptor of this T helper cell to the MHC2 molecule of the dendritic cell. This binding will confirm that this T helper and this dendritic cell are the cells of the same person, genetically identical. T helper cells, after this recognition by stimulation by the antigen, will replicate. You understand? They will proliferate, and during this proliferation, descendants of the T helper cells may be matured into the different types of the T helpers. But here shown T helpers type 1, T helper type 2, T helper type 17. Let's start from the T helper type 2. T helper type 2, when matured, secrete interleukin 4. This is a cytokine of these helpers. Interleukin 4 stimulates B lymphocytes that before recognized by their immunoglobulin receptors foreign antigens. It means this B cell has to be activated by foreign antigen, specifically where membrane-associated antibody contact to these antigens, and by the interleukin 4 cytokine or lymphokine stimulation of the T helper type 2. What this green B cell in our scheme is shown as the cell in the upper left corner of this drawing. This cell is activated by antigen, but this blue, activated by interleukin 4 of T helper type 2, These signals make the cell of maturation to the plasma cells. Plasma cells don't replicate, they short-living, live only 2 days, but during these 2 days they secrete huge amount of the molecules of antibodies. They are soluble antibodies, not the membrane secreted, soluble. These antibodies present a high concentration in the blood. Their presence demonstrate state of the immunity, and these antibodies interact with the antigens of bacteria of the type of that stimulated this dendritic cell from the beginning, and these antibodies bind to the bacteria, block the adhesins, attract the complement molecules, attract phagocytes, and help them to kill these bacteria. Finally, bacteria disappear. What is happening if other helper cells are activated? For example, if T helper cell is produced as a result of maturation of the antigen-activated T helper, these T helper cells secrete interleukin 2 and interferon gamma. Interferon gamma is the immunomodulator that facilitates phagocytotic activity of macrophages and antigen-presenting activity of macrophages. And macrophages start to work as the dendritic cell, also phagocytotically destroying bacteria and displaying antigens of these bacteria to other lymphocytes. And T helper 17, or this pink purple cell, T helper 17 produce interleukin 17 and produce interleukin 22. Interleukin 17 stimulates recruiting of neutrophils from vessels to the infected tissues. T helper type 2 causes so-called neutrophil inflammation that is characterized by infiltration of tissues by neutrophils. Neutrophils are very highly, very powerful killers of the bacteria. They are short-living, but during the short life, they may destroy many bacterial cells, cleaning the infected area from these cells. What is happened if dendritic cell presents bacterial virus antigen in the complex with MHC1 protein? Well, this MHC1 antigen protein complex stimulates not the T helper, but CD8 cytotoxic cells. And these cells replicate, and their progeny cells are transformed to cytotoxic T lymphocytes. These T lymphocytes produce perforin enzymes. They are shown in the right lower part of the slide, the white-blue balls that destroy the virus or bacteria-infected human cells that serve as the factories of producing the new parasites. And T killer cells that they should not individual bacteria or viruses, but should our change cells that are infected and maintain the replication of these pathogens. The next step, step number 5, destruction of non-self antigens and damaged self cells by immunocompetent cells, by different cells. So T killer cells, CD8, may destroy virus-infected cells. Macrophages may destroy, neutrophils may destroy. No, really, the stage is associated to produce of the such cytocidal substances that are hydrogen peroxide, nitric oxides, enzymes that destroy pathogenic organisms and our cells damaged by pathogenic organisms. The next, step number 6, excretion of the decayed products by excretory system of the human body. Excretion to remove destroyed or damaged tissues. Excretion via gastrointestinal system, urinary system, and respiratory system. And the last step of inflammation, step number 7, is associated to regeneration and recovery of damaged tissues. At this stage, post-inflammatory immunosuppression is happening. Immunosuppression is shown by the symbol that looks like the inverted T letter, not arrow. Arrow, the symbol of stimulation, but on this T, inverted T, symbol of the immunosuppression. And development of immunologic memories happen at this moment. Pay attention to this drawing. All the events in this drawing are subdivided into zones. So the zones, what barrier between these zones is shown by the dotted line, blue dotted line. All the events over this blue dotted line, the events characterizing active inflammatory process with the damaging of the own tissues, damaging of the bacterial cells. All the events lower this dotted line characterize process of inhibition on inflammation, process of the beginning of the regeneration, and beginning of the immunological memory formation. Now, very briefly, what number one in the top of this drawing? Inflammatory phase of the immune response. What do you see? You see the white arrow directed to the blood vessel, to the capillary, and you see that neutrophil migrates from the blood vessels to the infection area. Monocyte migrates, the T cells migrate. Monocyte in the tissues becomes mature to the M1 macrophage. No, two types of macrophages have been described: M1 macrophage and M2 macrophage. Both macrophages are progeny cells of the monocytes. The finally maturated form of the monocyte, these macrophages. In the process of active inflammation, of initiation of inflammation, monocytes are matured to the M1 macrophages. M1 macrophages produce pro-inflammatory signals. They secrete interleukin 1, B, interleukin 6, interleukin 12, tumor necrosis factors that stimulate changing of the blood flow and increasing of the vascular permeability, causing accumulation of liquid and leukocytes in the affected area. Additionally, M1 macrophages produce reactive oxygen species. What? Reaction oxygen species. For example, hydrogen peroxide, nitric oxide, what's shown here, or hypochloric radicals. All these radicals serve as a poison, destroying bacterial cells and partially destroying, causing lysis of the own damaged cells, damaged by bacteria, damaged by the immune response. It means that immune response in the form of inflammation is destructive for own tissues. It must be finished after disappearance of bacteria. When bacteria disappear, monocytes migrating from the vessels in tissues becomes mature to the M2 proteins. M2 proteins, what M2 proteins? The synthesis stimulated by interleukin 13 and interleukin 4 produced by T helper cells type 2. M2 macrophages produce interleukin 4 and insulin-like growth factors. They stimulate regeneration of tissues. M2 macrophages, what in the central part of the slide, they produce inhibitory signals that decrease synthesis of interferon gamma and tumor necrosis factor by cells that produce these pro-inflammatory cytokines. Interferon gamma usually is produced by T helper type 1 and tumor necrosis factor, but many cells, including macrophages. It means, well, this is an inhibitory signal. Additionally, M2 proteins secrete interleukin 10 and tumor and the transforming growth factor beta, but even these substances stimulate inhibition of the pro-inflammatory cytokine producing, and interleukin 10 transforming growth factor beta, both serve as the immunosuppressors. And these immunosuppressors produced not only by macrophage type 2, but T regulatory helper cells also. These cytokines stimulate accumulation of the fibroblast in the zone of inflammation and formation of the connective tissue to replace destroyed tissue, and stimulate the reparation process, remodeling of tissues, and finishing of the inflammatory reaction. What my task now will be tell you about some of the substances involved in this inflammatory process. So, innate immune factors can be classified. Now we'll say about innate immune factors to the physical barriers, mechanical factors, such as the skin cells, keratinocytes, mucous membrane cells, and normal microbial flora that lives in our intestine and struggles against pathogenic flora. Second group of factors, blood proteins such as the complement system and interferon system. Third group of innate immunity factors, cellular factors, macrophages, neutrophils, natural killer cells, dendritic cells. And the last one, the group of cytokines that regulate and coordinate activities of the cells of the immune system. You know that tumor necrosis factor, interleukin-1, interleukin-6, they are pro-inflammatory cytokines, enhancing active destroying of bacteria and their own infected cells. Skin. Skin consists of the many layers of keratinocytes. Upper layers consist of dead skin cells. They establish the serious depth barrier for bacteria. If bacteria or other parasites penetrate layers of keratinocytes, they find in the deep parts of the skin Langerhans cells. Langerhans cells recognize foreign pathogen-associated molecular patterns of bacteria, engulf these bacteria. After that, start migration through the lymph nodes to the lymph tissues, lymph nodes, for example. When these cells come to the lymph nodes, they are transformed to the dendritic cells that present antigens of the engulfed bacteria to the lymphocytes, T lymphocytes, T helper cells that occur in the subcutaneous tissues and activate macrophages, T lymphocytes after their stimulation. And from this moment, inflammatory process is initiated. Mucous membranes. Mucous membranes consist of the mucous membranes cells in the respiratory epithelium, the ciliated cells. Between the cells in the intestinal system occur so-called macrofold cells. Macrofold cells are characterized by the having of the large pockets in the part of cell that is exposed to the basal membrane of epithelium. Function of M cells, they maintain a very slow process of the engulfment of the bacteria, transporting of these bacteria through the cytoplasm of the macrophage cells, and presenting these bacteria to the macrophages and dendritic cells in the pockets. These cells destroy bacteria and present antigens to the lymphocytes, to T lymphocytes. B cells also react to the antigens of these organisms. These lymphocytes are transported to the submucous lymphoid tissues, where the cells, B lymphocytes, produce immunoglobulins A, secreting immunoglobulins A. They are secreted to the lumen of the tract, intestinal tract, or respiratory tract, and bind to the antigens of bacterioviruses, preventing binding of these bacterioviruses to the human cells, preventing in this way beginning of the infection. Now about complement system. Complement system is the system of proteins that circulate in our blood in the inactive state. in normal conditions. Complement system consists of more than 26 heat labile proteins. These proteins are activated in the process of the recognition of the non-self microbial components and cause at least 4 effects. Activation of complement system causes lysis of bacteria and other cells. In the photograph you see hole made by C9 complement proteins that form the walls of the channel, pathological channel for bacteria or for human cell, and through these channels bacteria or attacked human cells lose the water and ions and die due to this. This process is named lysis, bacteriolysis, for example, if bacteria lysed. Second biological effect, production of chemoattractants for phagocytes. And chemoattractants, they are substances that, when are produced, make the migrating cells moving to the area with a high concentration of chemoattractants. If complement system is activated in the infected area, of course, these chemoattractants stimulate recruiting of the leukocytes from blood vessels to the tissue, accumulate, that is, facilitate accumulation of them in the tissues. Third effect, generation of mediators of inflammation. Mediators of inflammation, for example, histamine, that increases blood flow in the vessels, and in this way increases the vascular permeability and the accumulation of liquid and immune cells in the infected area. And the last effect, opsonization. Opsonization, this is the effect of the enhancement of phagocytosis of bacteria that are coated by components of complement system that are named opsonin. No, the main opsonin of complement system is C3b protein. Different complement activation pathways are known, but eventually all these pathways of activation converge to make the same end products. Here you see a scheme of the 3 pathways of activation of complement system. Classical pathway, left, lectin pathway, middle, and alternative pathway, right. First discovered and most powerful is classical pathway. Pay attention, at the top of the classical pathway you see triangle antigen of bacteria and Y-shaped antibody, immunoglobulin G antibody or immunoglobulin M antibody. This antibody react to the antigen, forming antigen-antibody complex. This antigen-antibody complex stimulates C1 component or C1 fraction of the complement system. This is the first protein activated. This protein consists of 3 types of the subunits, C1q, C1r, and C1s. After activation by antigen complex, this C1 protein cleaves, activating in this moment C4 molecule and C2 molecule of complement system. C4 and C2 molecules are coded for C4a, C4b, C2a, and C2b subunits. C4b subunit, isolated from C4 protein by the C1 protein, binds to the C2a component released from the C2 component complement system, and you see formation of 2 component complex, C4b one component, C2a second. This complex is named C3 convertase. It means that C4b to A complex, this newly formed molecule, serves the enzyme that cuts C3 molecule of complement system for C3a and C3b, C3a and C3b component. C3b component binds to the C3 convertase, forming 3 component complex, C4b to A3b. This complex serves a new enzyme that is named C5 convertase. C5 convertase cleaves C5 molecule of complement system with the production of C5a and C5b subunits. C5b subunits, when it's synthesized, become sticky for lipid membranes of the human cells or bacterial cells. C5b binds to the surface of the bacterial cell, attracts C6, C5b6 complex is formed. This complex attracts to the C7, C8, and about 10 molecules of C9. As a result, complex C5b 6, 7, 8, and 9 is formed, and this complex is named membrane attack complex. This complex penetrates all the depths of the lipid membrane. Walls of this perforation walls are formed from C9 proteins, about 10, 9 or 11 molecules. And this tube, pathological pore, can be closed by this bacteria, by the human cell. Becomes open through this whole ions are released, water is released, is loosened, and bacterium or human cell dies. This is a classical pathway. To initiate classical pathway, we have to see antibodies in the human body. Before synthesis of antibodies, at the beginning of the inflammation, other pathways may be realized. Second pathway is lectin pathway. How is initiated this pathway? If many bacteria have the carbohydrate polymers with the terminal mannose residue. If the carbohydrate chain has the terminal mannose, mannose binding lectin, this may be soluble or mannose binding lectin on the surface of some immune cells, interact to this mannose. And this lectin becomes activated, and after that cleaves C4 and C2 molecules as is made by C1 molecule in the classical pathway. It means mannose binding lectin activated by terminal mannose of bacterium cleaves C4 and C2 molecules for CA and C subunits. After that, all the events up to the or down to the formation of membrane complex are the same. Third pathway is alternative pathway. This alternative pathway ignores presence of C1, C2, and C4, but involves in the process of activation complement proteins B, D, and P. What has happened? Usually in the normal life, C3 molecule of complement system, after the finishing of the molecular life, start to be degraded by enzymes, and C3 is subdivided into C3b and CA molecules. Usually very quickly these molecules ruptured and disappear. But if in the moment of the formation of C3b molecule, near to this molecule occurs bacterium with the lipopolysaccharide, for example, gram-negative bacteria have lipopolysaccharide, Escherichia coli, Shigella, Salmonella. Lipopolysaccharide binds to C3b and protects the C3b from hydrolysis by protease that in other conditions could be destroyed. C3b molecule. In the presence of lipopolysaccharide, C3b remains stable. Well, C3b after that binds B protein of the complement system. C3b B complex is formed. After that, B capital. After that, D protein will interact with this two-component complex and will form C3b B large, B small. It means B large, B small. This is a B component of the B protein of the complement system. With this C3b B serves the C3 convertase of the alternative pathway and destroys C3b to C38 and C3b. It means C convertase C3 convertase of the alternative pathway and classical pathway, their formula are absolutely different, but effects course are same. After that, C3b component of the CRIF C3 molecule is shaded to C3 convertase, forming three-component complex that is named C5 convertase, and C5 is activated after C5b, is involved in the membrane complex formation, and complex is formed. We told you that there are more than 26 complement proteins are shown, but here you see 9 types of the C molecules and 1, 2, 3 types of BDP. It means 12 molecules. What about other 14? Other are shown as the inhibitors of this system. For example, in the classical pathway, CD55, CRY, CD46 molecules serve as the membrane molecules of human cells that inhibit formation of the C5 convertase of the classical pathway. This is a complex of down-regulating molecules that inhibit further development of complement activation after disappearance of the bacterial antigens. The membrane complex synthesis is prevented by CD59 complement protein. C5 convertase of the alternative pathway is blocked by factor I and factor H, also membranous molecule of complement system. It means you see here not only mechanism of activation, but mechanism of the inhibition of activation of complement system that is required after disappearance of the bacteria or other irritating factors, foreign factors influence to the activity of the complement system. But in this slide you see classical pathway, or C1, C4, and other bacterial lysis. What do you see here in the central part of the drawing? You see that C3A and C5A small soluble molecules that synthesize in the process of activation of complement system may serve the chemoattractants for the leukocytes that are attracted to the infected area. Also, these C3A, C5A molecules may stimulate, may make mast cell of releasing histamine. Histamine stimulates the blood flow, stimulates the increase in vascular permeability, enhancing infiltration of tissue by the water and leukocytes. And C3b molecule of complement system is itself may bind to the bacteria and may stimulate engulfment of this bacteria by phagocytes. This stimulation is named opsonization. And due to this, C3b component of the complement system is itself is named by the word opsonin, substance capable of enhancing phagocytosis. Second system is interferon system. Interferons, in the contrast to the other soluble proteins of the innate immune system, are host species specific. It means human interferons may act only in the human organism and will demonstrate no effect in the any animal organism. All the interferons are subdivided in 3 subfamilies: in alpha interferon, beta interferon, and gamma interferon. Alpha interferon, leukocyte interferon, inhibits growth of viruses by blocking the translation of virus proteins. Beta interferons also have antivirus and limited anti-cancer effect. Gamma interferon serves as the immunomodulator. It means gamma interferon may stimulate formation of the M1 macrophages, so transformation of the monocytes to the M1 macrophages, and gamma interferon prevents transformation of the monocytes to the macrophage type 2. Interferon gamma stimulates phagocytotic reaction of macrophages. Interferon gamma stimulates formation of T helpers type 2 and prevent maturation of T helpers type 2. Immunomodulator means not only stimulating substance, but substance that may stimulate some processes and inhibit at the same time other. Three cytokines are shown in this slide. For example, first, tumor necrosis factor, produced by macrophages T cells. Why? Because these macrophages T cells recognize foreign molecules on the surface of the bacteria that are named pathogen-associated molecular patterns. After recognizing of these patterns, tumor necrosis factor is produced and causes following effects. Activation of endothelial cells for inflammatory process and coagulation. For neutrophils, activation of neutrophils for the phagocytotic reaction. Hypothalamus, stimulation of fever. Tumor necrosis factor tracking to the hypothalamic cells increases the body temperature. Tumor necrosis factor stimulates in the liver synthesis of so-called acute phase proteins. Acute phase proteins, they're proteins that are very low concentrated in normal conditions, but their concentration increased about 1,000 times during the inflammation. What? During the synthesis, as the result of the activation of tumor necrosis factor, the synthesis of these acute phase proteins in the liver. What are the major effect of these proteins, acute phase proteins? They limit spread of microbes or microbial products in the host organism. The most important proteins of this group are C-reactive protein, alpha-1 antitrypsin, alpha-2 microglobulin, fibrinogen, serum amyloid A, and other. Exception for what these activities, tumor necrosis factor also causes cachexia. It means losing of the mass of the muscle cells and fat. And they stimulate apoptosis. This is a suicidal activity of many cells involved in the inflammatory process. Analogs effects are caused by interleukin-1 and interleukin-6. Exception for macrophages, interleukin-1 and interleukin-6 may be secreted by endothelial cells and some lymphocytes, for example, T lymphocytes. Cellular factors of the innate immunity are phagocytotic cells. They're subdivided to macrophages. They're long-living cells. Long-living means they live from 7 to 50 days. And macrophages, short-living from 5 to 12 days. Macrophages, you know, are descendants of the monocytes, and macrophages may serve as the antigen-presenting cells because they have MHC protein type 2, major histocompatibility protein type 2. Macrophages are only phagocytotic cells. They are neutrophils, eosinophils, and basophils, so they cannot serve as antigen-presenting cells. Activity of the macrophages may be regulated by chemokines. Chemokines is a brief form of the term chemoattractant cytokines. Chemoattractant cytokines, they are small polypeptides, about 10 kilodalton, the molecular weight, that are produced by many cells, local endothelial cells, epithelial cells, fibroblasts, resident neutrophils, and macrophages. All these cells produce cytokines, in this case chemokines, as the chemoattractants, after activation of the fibroblasts and other cells that are listed here by the pathogen-associated molecular patterns or damage-associated molecular patterns that serve as the markers of the foreign non-self. Cytokine production, inflammatory cytokines, tumor necrosis factor, interleukin-1. There are 4 types of chemokines have been described. 2 of them are major: CC chemokines and CXC chemokines. CXCL9 and CXCL10 chemokines, when are secreted, they are secreted by macrophage type 1, and when are they secreted, these macrophage type 1 cytokines, chemokines, stimulate activation of T helpers type 1. It means they stimulate process of the maturation of naive T helper 0 to the T helper cell 1. T helper cell 1 secrete interferon gamma, we told about this, and interferon gamma activate phagocytotic activity of macrophages and facilitate antigen-presenting properties of the macrophage type 1. Signal for secretion of the chemokines that are shown here, CXCL9 and L10, is the result of the recognition of toll-like receptors, the pattern recognizing receptors, of the pathogenic-associated patterns such as the lipopolysaccharide of bacteria. Also, M1 proteins, by production of interleukin 12, activate T helper 1, activate natural killer cells. In the contrast, CXL17, CCL17, CCL22, and CCL24 are produced by macrophage type 2, or this anti-inflammatory macrophage that is becomes prevalent in the face of the immunosuppression. These macrophage type 2 chemokines stimulate activity of T helpers type 2. Stimulated T helpers type 2 produce interleukin 33, interleukin 4, interleukin tumor necrosis factor also, that stimulate maturation T helpers type 2. Also, basophils and innate lymphoid cells may stimulate transformation of monocytes to the macrophage type 2. It means you see that chemokines produced by different macrophages activate different types of cells involved in the immune response. Phagocytosis may be subdivided in the subphases of migration, adhesion, ingestion, and digestion of the pathogens. Adhesion of phagocytes is initiated by interaction of the integrins of the macrophage surface to the selectins, ECAM and VCAM molecules that are situated on the endothelial cells. Adhesion and migration of macrophages to the tissues from the vessels is the result of the binding of integrin to the selectins, where concentration of selectins becomes high. This enhancing of the concentration of the endothelial cells is the result of the activity of the chemoattractants. Digestion of bacteria and other phagocytotic objects by macrophages is caused by 2 oxygen-dependent killing and oxygen-independent killing mechanisms. Antigen-dependent killing mechanism is a result of so-called respiratory burst and is realized in the quick and powerful release of superoxide radicals, O2 with a minus charge, hydrogen peroxide, hypochlorous, and nitric oxide radicals. These oxygen-dependent reactive species, oxygen-containing species, oxidize components, proteins, carbohydrates, and lipids of the bacteria, causing them partially destroyed, and lysosomal enzymes, complex oxygen-dependent killing mechanisms, very quickly destroy these bacteria that are phagocytosed or occur near to the phagocytes. These actually independent enzymatic killing substances are lysozyme, lactoferrin, cationic protein, proteases, nucleases, and lipases. How innate immunity cells recognize foreign molecules? Here you see dendritic cell that engulfs the bacteria because bacteria have markers of non-self in the form of the pathogen-associated molecular pattern. These patterns may be recognized by pattern recognizing receptors of the leukocytes that occur on the surface of these cells, the membranes. Primary recognition of non-self is based on the reaction to the non-self molecules or damaged self molecules. Non-self molecules are named pathogen-associated molecular patterns, PAMPs. They are double-stranded and single-stranded RNAs, bacterial, unmethylated cytidine guanine nucleotides, formyl methionine, pillin, collagenine, lipopolysaccharide, lipoteichoic acid, and many other substances. They all are named pathogen-associated molecular patterns. They are markers of foreign molecules, but they are not antigens, because these substances are not recognized by antibodies. Second group of the irritating for innate immunity are damage-associated molecular patterns, DAMPs. They are endogenous molecules produced by damaged cells. For example, stress-induced proteins such as the heat shock proteins, monosodium urate crystals, nuclear proteins, fibronectin, fibrinogen, and some other molecules. What the receptors of our cells may recognize these PAPs and DAMPs. They're different molecules. All of them are named pattern recognizing receptors, PRR. PRR are localized on the various cell types. The dendritic cells are the phagocytes, B cells, endothelial cells, epithelial cells. The most studied and understandable pattern recognizing receptors are Toll-like receptors, node-like receptors, and Rig-like receptors. Node-like receptors and Rig-like receptors may occur in the cytoplasm. Toll-like receptors mostly are membrane-associated receptors. Here you see list of the different 11 Toll-like receptors. You see that they react to the different bacterial, virus, human compounds. For example, Toll-like receptor 4 recognizes such pumps as the lipopolysaccharide, F protein of the respiratory syncytial virus, some human components, human heat shock protein 60 and heat shock protein 70, the macromolecules of the proteins, fibronectin, fibrinogen, oligosaccharides of hyaluronic acid of the human origin. Toll-like receptor 6, for example, except for bacterial lipoproteins, react to the pump in the form of fungal substances. Toll-like receptor 7 and 8 may recognize single-stranded RNAs of the virus origin. How to work these molecules? Toll-like receptor type 1, with this green, this is a cell. Toll-like receptor number 1. Toll-like receptor type 4, for example, react to the lipopolysaccharide. After that, second molecule of toll-like receptor 4 binds to this lipopolysaccharide also, causing dimerization of two toll-like receptors type 2. It causes juxtaposition of the tear domains, cytoplasmic portions of these receptors. These tear domains initiate a signal that transports information about this lipopolysaccharide recognition to the so-called nuclear factors, for example, nuclear factor kappa B, TRF, and other. These nuclear factors, after activation in cytoplasm, migrate to the nucleus of the cell, binds to the certain specific portions of the DNA, and facilitate dispersation of these DNA molecules and transcription from them. Transcription causes translation, synthesis by this cell of the proteins encoded by these DNA fragments, for example, inflammatory cytokines. Here you see a more complex scheme. You see dimerization of toll-like receptors reacting to the same lipopolysaccharide. Tear domains are shown brown. In the cytoplasm occur so-called adapter proteins, they are shown here by the blue color, that also have tear domains. Tear domains toll-like receptor 4 interact to tear domain of the MID88 or TRIF, and these proteins transfer signal to the second group of enzymes that is named protein kinases, for example, Iraq proteins. But even these protein kinases activate nuclear factor kappa B, TRFS, to the stimulation of expression of genes responsible for immune response. NF-κB stimulates synthesis of the such inflammatory substances, the cytokines, tumor necrosis factor, interleukin-1, interleukin-6 in the macrophages, of the chemokines, CCL2, CXL8, endothelial adhesion molecules, selectins, co-stimulating molecules CD80, and many other. All these molecules cause acute inflammatory process. Other nuclear factor, TRFS, stimulate expression of proteins encoded by the genes and that code for interferon alpha and interferon beta, these antiviral substances. In this scheme, you see more detailed scheme of the involvement of toll-like receptors with this, and this, in the intercellular signaling pathway that is finished by the activation of synthesis of pro-inflammatory cytokines, for example, by the genes if they inculcated by an NF-κB factor. Natural killer cells. Natural killer cells don't engulf bacteria, but they kill bacterial virus-infected cells by extracellular killing. They are lymphocyte-like cells which are present in active state prior to antigen stimulation and are not specific for antigens. Natural killer cells kill in normal cells by secretion of perforins and granzymes. The substances causing perforations in the cell membrane, and granzymes pass through this membrane through these perforations, causing hydrolytic destroying of the internal structures of the infected bacteria. The activity of greatly increased by exposure of the interferons and cytokines. How these processes happen? What natural killer cells? 3 cells are shown on the left, part of the couple, and healthy or target cells on the right. What in the upper? What we have to know? Natural killer cells distinguish infected and stressed cells from healthy cells, and their activation is regulated by a balance between signals that are generated from activating receptors. Activating receptors are shown green, activating receptor natural killing cells. Balance between activating cells and inhibitory receptors. Inhibitory receptors are orange. Inhibitory receptors interact to the major histocompatibility class 1 proteins. What do you see here in the upper part? You see interaction of cell. Cell has 2 MHC class 1 molecules that activate 2 inhibitory receptors. Natural killer cell has 1 activating receptors that binds to the ligand of stimulating receptor. Totally, you see 2 inhibitory signals through the inhibitory receptors and only 1 stimulating receptors. Number of inhibitory receptors overlay stimulating receptor signals, and the cell, healthy cell, is not attacked and not destroyed. Lower. We see other situation. We see that natural killer cell contrasts to the tumor cell, shown blue. Tumor cell has 1 activating receptor reacting to the activating ligand on the surface of tumor cell, and this tumor cell doesn't have correctly presenting MHC1 proteins. It means inhibiting signals are absent. In this case, natural killer cell secretes cytotoxic molecules, perforins and enzymes, and kill tumor cell. In the lowest part, what do you see? You see natural killer cell that recognizes 2 MHC1 molecules on the surface and generate 2 inhibitory signals. But tumor cell here has more number of the activating receptors, more ligands for activating receptors, and activating receptors generate 3 signals of activation, and this cell has only 2 signals for inhibition. Of course, the cell, natural killer, will attack the tumor cell, killing this cell. What's the simple scheme of this you see here? Inhibitory receptor shown in green. Some of these receptors are NKG2A and NKG2B. They're inhibitory receptors. They're ligands of the MHC1 protein. Healthy cell may stimulate, may expose many ligands for activating receptors, but if number of the activating signals by activating receptors and inhibitory receptors will be the same, healthy cell will be also free from attack. If number of activation receptors is higher than number of inhibitory receptors, infected outer Lego cell becomes attacked and destroyed. Some words about acquired immunity. Acquired immunity is based on the recognition of the non-self molecules named antigens. Antigens may react to the T cell receptors of T cells and immunoglobulin receptors of B lymphocytes. But here you see molecule of antibody reacting to the virus antigen by the antibody antigen binding sites. Here you see 2 sites, antigen binding sites of this antibody. There are at least 2 attributes of antigens. Really very many, but let's discuss only 2. One is antigenicity and second is immunogenicity. Complete antigens must be antigenic and immunogenic. What does it mean, antigenicity? This is specific reaction of the antigen to the antibody or sensitized cells, or to the TCR receptors of sensitized cells. What is immunogenicity? This is a capacity of induction of immune response. It means antigenicity, this is a reaction of antigen to the performed antibody. Immunogenicity, this is a stimulation of the synthesis of antibodies. Antigens may be classified in 2 different types, haptens and complete antigens. Haptens are not immunogenic antigens. Many antibiotics are known as the haptens, on incomplete antigens. For example, penicillin, we see here molecules of penicillin. This is a rather low molecular weight substance, and due to this, penicillin cannot activate synthesis of antibodies and is not immunogenic. But if antibodies occur against penicillin, penicillin will bind to these antibodies. Haptens are not immunogenic because they can't activate T helper cells. due to inability of haptens to bind to the MHC proteins. Do you remember? Major histocompatibility proteins. They present complete antigens to the T cells. Haptens can bind to the MHC. Haptens can activate B cells that can stimulate immune response by themselves. Complete antigens. They induce immune response, first immunogenetic, specifically react with antibodies and activate immune cells. It means that complete antigens must be enough large. Molecular weight of these proteins usually has to be not less than 10,000 daltons, 10 kilodaltons. For example, haptens may have weight lower than 1,000 daltons. Immunogenicity. It means if antigen is inoculated to the human or animal body, initially this antigen may be recognized by only a few cells that have membrane receptors for this antigen, specific for this antigen. But after recognizing of this antigen by the cell, T cell, for example, B cell, what here B cell you see. B cell start replicating, and initially some of these descendants of these B lymphocytes are transformed to the plasma cells and produce immunoglobulin M. In the graph characterizing levels of immunoglobulin, you see a small peak of the immunoglobulin M. Other cells repeatedly replicate and transform to the plasma cells later. They form the immunoglobulin G that is shown as the red peak. Totally green peak demonstrates the total concentration of antibodies. Some of the descendants of the initially activated by this antigen B lymphocyte, some are transformed to the memory cells. They are not mature to the plasma cells and have the resting cells in the lymph system. If the same antigen will enter to the same organism again, this antigen will contact not to few but to many memory cells, and memory cells will continue process but at the higher level. Immunoglobulin M peak would be the same, but immunoglobulin G peak would be very high and long-lasting. Haptens may be transformed to the complete antigen if haptens chemically would be bounded to the carrier protein. Small molecular weight hapten in the complex with the carrier protein becomes transformed to the complete antigen. Antigenic determinants. Antigenic determinants are the smallest units of antigenicity, smallest. They react to the different antibodies that have different antigen binding sites. If you will study glycoprotein spike of the virus, you will see protein. The analysis of this protein will show this protein consists of the tertiary conformation of the protein of the polypeptide. This conformation has the special elements that are shown by different colors that may be recognized by different antibodies with different specificity. But every of these sites of the structure of the antigen that may be recognized by the monoclonal antibody is named epitope or antigenic determinant. This antigenic determinant must react to the antigen binding sites of the immunoglobulins, of the antibodies. So this immunoglobulin G has these two antigen binding sites that are marked by letters V. Every antigen binding site consists of the fragments of the two chains, purple and green. Purple and green chains in the right part of the slide are shown as the elements forming specific pocket. If certain antigenic determinant has the external configuration of atoms that spatially fits to the configuration of the pocket formed by the atoms of the variable parts of the immunoglobulin that form antigen binding site, you will see very highly specific interaction of antigen and antibody. Antigen in this case fits to the antibody as the key to the lock. Human cells have many antigens. Majority of these antigens are marked by the symbol CD and number. CD means clusters of differentiation. What you see here, B cell and T lymphocyte. Majority of the antigenic determinants of the cells have these receptors. For example, T lymphocyte has either CD4, either CD8 receptors. They never occur on the surface of other cells, only T helper cells, CD4, or T killer cells, CD8. CD3 receptor is intimately bounded to the T cell receptor and forms one complex that T cell recognizes foreign antigen, a signal about this recognition through the CD3 receptor transformed it to the genome of the cell. In the B cells, typical for the CD are CD19, CD20 receptors, CD40 receptors, CD5 receptors. Immunoglobulin receptor for antigen of B lymphocytes is immunoglobulin M, with this purple. Antigen receptor of the T lymphocyte is TCR, not immunoglobulin M receptor. In the B lymphocyte, you may see some molecules that are not named as the CD. For example, MHC2 molecule. This is a marker of all the antigen-presenting cells. Now, I wouldn't concentrate my mind to this. Pay attention to this slide. Acquired immune response may occur in different forms. For example, in the form of the humoral antibody-mediated immune response, or in the form of the cell-mediated immune response. When antigen enters the human body, we know this antigen may be engulfed by the antigen-presenting cell, dendritic cell or macrophage. Dendritic cell or macrophage presents antigen to the helper T cell. Helper T cell may produce and must produce certain cytokines. If helper T cell is the helper T cell type 2, this T helper cell secretes interleukin 4, interleukin 5. These interleukin 4 and 5 cytokines, they stimulate B cell that before was activated by the antigen separately. It means B lymphocytes via immunoglobulin receptor react to the antigen, and the T helper cell stimulated by the antigen via interleukin 4 activate replication of B cells. B cells replicate. Some of them are transformed to the plasma cells that produce antibodies. Well, these antibodies are markers of the humoral antibody-mediated immune response. Antibody synthesis. Some B cells becomes conserved as the memory cells. Helper cells also, some of them are transformed to the rested cells, memory T cells. What about cell-mediated immune response? The cell-mediated immune response, antigen-presenting cell stimulates, interacts to the cytotoxic T cell, CD8 cell. And if helper type 1 cell is activated, this T helper type 1 cell produces interferon gamma that stimulates accumulation of cytotoxic T cells. Some of these activated cells will be transformed to the cytotoxic cells that will attack and kill foreign virus or bacterial infected human cells, and some will be preserved as the memory cells. Antibody synthesis is the result of the cooperation of antigen-presenting cells, these helper cells, and B lymphocytes, Antigen-presenting cells The phagocytes that engulf bacteria or viruses isolate from these bacteria antigenic peptides, combine these peptides to the MHC2 molecules that are shown by the blue bricks here, and in this form are presented to the helper cells. If we see about antibody synthesis, these cells, first of all, helper cells are T helpers type 2. Also, T helpers type 2, exception for activation by the antigen presentation, becomes activated by interleukin 12 and 1 secreted by antigen presenting cell. T helper cell replicates from naive helper cells mature to the T helper type 2, and the cell produces high concentration of interleukin 5 and 4. These interleukins stimulate B cells, but not all. Only B cells that before could recognize antigenic determinant by the immunoglobulin membrane receptors of B cell. It means B cell activated by antigen may acquire interleukin 4 and interleukin 5 humoral signal. And this B cell must react to the T helper 2 cell via CD4 core receptors that must react to the CD4 ligand receptors of T helper 2 cells. It means what only after this three-component stimulation by antigen directly via immunoglobulin receptor, stimulation via interleukin 4 and 5 of T helper type 2, and stimulation by core receptors, CD40 B cell receptor react to the CD40L, what this B lymphocyte will proliferate, will be transformed mostly to the plasma cell that will produce antibodies. A part of these cells, of these progeny cells, would be stopped in their maturation in the form of the memory B cells. Now, here you see how dendritic antigen-presenting cell recognizes foreign object, a virus or bacteria, because pathogen-associated molecular potential of these bacteria react to the two toll-like receptors that initiate cascade of reactions in the signaling pathway that activate nuclear factor kappa B, that stimulates synthesis of the co-stimulatory and adhesion molecules and cytokine production. Antigenic peptides isolated from these bacteria combine to the MHC2 molecules that are shown green here. These molecules combine to the antigenic peptide in the Golgi apparate. After that, these complexes, MHC2 antigenic peptide, are exposed on the surface of the antigen presenting cell and displayed to the T cell receptors of the T cell, in this case T helper cell. After this, this T helper cell becomes activated. Here you see structure of the T cell receptor, so-called alpha beta T cell receptor. This T cell receptor of T lymphocytes recognize this signal, recognize this antigen, but a signal about this recognition must migrate to the CD3 receptor, epsilon delta and gamma epsilon CD3 receptor that has the transmembrane portion that may initiate signaling pathway, stimulation of the synthesis of the immune molecules by the cell after recognition of T cell receptor. Some T cells have gamma delta T cell receptors. What is this? This, in the contrast to alpha beta lymphocytes, gamma delta TCR, in the contrast to the alpha beta TCR cells, gamma delta TCR has the less TCR diversity. It means diversity of the antigen that may be recognized by these receptors. They are enriched at epithelial surfaces and appear to respond to self molecules that signal potential danger or cellular stress. tumor cells, for example. They have shown anti-infection, anti-tumor, and immunoregulating activity. What do you see here? You see here a process of activation of B cell, activation of T cell. First stage. B cell, you know that this is antigen presenting cell, has surface immunoglobulin molecules. If these molecules specifically react to the antigen that's shown in the blue, signal about this recognition, especially to the nucleus, and the cells start to produce MHC class 2 molecules, with NCD4, 40 molecules. As a result, after processing, digestion of microbe and processing of antigen, peptides, antigenic peptides of this antigen are combined to the MHC class 2 antigen that is presented by this B cell. After that, antigen presentation to CD4 helper cell is happened. So this B lymphocyte presents antigenic peptide in the complex to the MHC2 molecule to the T cell receptor of the T helper cell, naive T helper cell. Additionally, CD4 receptor of helper cell reacts to the MHC2 molecule to confirm that this MHC2 molecule and all these B lymphocytes legitimately react to the T helper. For CD4 contact to MHC2, this is a signal of confirming that both these cells are cells of the same human or animal organism. Additionally, CD40 ligand of B cell react to the CD40 ligand molecule of T helper. And the signal about this interaction also stimulates nucleus of the B lymphocyte. Additionally, third stage, stimulation of T cell, and stimulation of T cell in the synthesis of interleukin 4 and 5. Activated T helper cell, T helper type 2, when is stimulated via TCR receptor by antigen, via CD4 receptor that contact to the MHC2, and via co-receptor with CD40 ligand of the helper react to the CD40 of the B lymphocyte. Activated helper cell secretes interleukin 4 and 5. They are recognized by specific receptors for these substances, and this is a last activating signal that initiate replication of this B cell and transformation of B cell to the plasma cell to antibody synthesis. So, to initiate synthesis of immune reaction, if initiate immune reaction, what is happening? So, presentation of antigen must occur. In the right part of the slide, you see activation of helper T cell, or this upper helper T cell, CD4 cell, by the antigen, shown red, MHC2, or this orange, protein complex. Here you see presentation that is directed for synthesis of antibodies. So T cell receptor of the T helper cell consists of two subunits. Two subunits specifically react to the antigenic peptide that is presented by two subunits of MHC2 molecule. After that, CD4 receptor of T helper cell interacts to the one of the subunits of MHC2, confirming that this recognizing of antigen is correct. After that, signal that recognition is correct is transformed to the CD3 molecule, and from CD3 molecule, by the signaling pathway, information comes to the nucleus, and T helper cells start to be activated. Other example of the antigen presentation: when MHC1 molecule of the target cell is associated to antigenic peptide. Activation of in this case, not helper, but cytotoxic CD8 T lymphocyte is activated. This T lymphocyte may kill target cell that is shown here. It means TCR of the activated cytotoxic cell recognizes antigen presented by one polypeptide transmembrane chain of the MHC1 protein. MHC1 protein occurs in all nucleated cells, MHC2 only in the antigen present. After this recognition, CD8 receptor must react to the MHC1, confirming that this somatic cell the somatic cell belongs to the family of cells, to that belongs to this T cell. It means cells of the same organism. After that, signal is transformed from the TCR to the CD3, and after that to the nucleus. What is happened in the presentation of antigen in the synthesis of antibody? Microbe is phagocytosed, is transported to phagolysosome. Phagolysosome enzymes destroy microbe, and release of this microbe triangle antigenic peptides. At the same time, in the endoplasmic reticulum, MHC2 molecule is synthesized together to the CLIP peptide that closes the antigen binding cleft of the MHC2 molecule for a certain time. After that, from the endoplasmic reticulum, this MHC2 molecule, the membrane molecule, is transported to the Golgi apparate. In the Golgi apparate, antigenic peptide from the microbe engulfed before replaces with this clip blocking fragment and becomes situated in the antigen binding cleft of the MHC2 molecule. And finally, this MHC2 molecule, together with bacterial antigen, is exposed to the surface of this cell. Because this is MHC class 2 molecule, we say about further synthesis of antibodies. It means critically important type of the antigen presentation to understand what the immune response would be developed. This is the last slide. In the right part of the slide, you see exogenous antigen presentation. In the left, in the green zone, presentation of endogenous antigens. Exogenous antigen is presented as the antigenic peptide in the complex to the class 2 MHC molecule. Endogenous antigen, antigen of the virus that replicates in the cell, is presented on the surface of this antigen in the complex to the MHC class 1 molecule. What has happened when exogenous enzyme, exogenous presentation is happened? Initially, in the rough endoplasmic reticulum, MHC class 2 molecules synthesized together with yellow invariant chain. After that, this invariant chain becomes lysed, and we see that MHC2 molecule has only small clip fragment that is situated in the cleft that in the future would be devoted to localize here bacterial antigenic peptide. This cell is a macrophage, the dendritic cell, engulfs phagocytotically bacterial or virus protein. This bacterial virus is disrupted in the phagolysosome, and after that, for this exogenic peptide, contrast to the MHC2 molecule, clip fragment is removed by the excessive concentration of the antigenic peptides, and this peptide, one of them, binds to the MHC2 molecule in the form of the MHC2 antigen molecule complex that is exposed on the surface. In the contrast, in the endogenous presentation, protein of foreign origin is synthesized by the parasite that lives inside of this target cell, this infected cell. Endogenic peptide of the virus as the own peptide is undergone to the destroying the proteasome. Proteasome cuts this endogenic antigen for the antigenic peptides. These peptides through the TAP pore system is injected to the rough endoplasmic reticulum and is exposed to the precursor of MHC1 molecule in the process of the maturation of this molecule. And in the process of maturation, binds to the antigenic binding cleft of MHC1 protein, and in this form is transported from the rough endoplasmic reticulum to the Golgi apparatus, from Golgi apparatus to the surface of bacteria. And exogenous antigen exposed in the complex to the MHC2 protein, endogenous antigen, the complex to the MHC1 protein. Class 2 MHC protein antigenic peptide is recognized by T helper cells. Complex MHC class 1 to the antigenic peptide is recognized by T killer cells, CD8 cells. This is the last slide that I want to see you. I hope that you will understand this topic better after paying attention to this lecture. Thank you for your attention. Goodbye.